Okay, so I will start in the troubleshooting lecture, and this lecture is about immunostaining, the usual problems and tips on how to avoid them. I divided my lecture in 3 parts. The first part I will give a short introduction about how immunostaining works. In the second part, I will talk about, give you some tips on where to look for antibodies and what antibody validation is all about. And the third part, I will talk about immunostaining, the controls, and I will highlight some of the immunostaining steps we perform and what to pay attention to. So let's start with the first part of my lecture. So when we are talking about immunostaining, we quite often encounter the words immunofluorescence, which is abbreviated IF, immunohistochemistry, which is abbreviated IHC, or immunocytochemistry, ICC. They are all referring to immunostaining. So if you just use the word immunostaining, you will never go wrong. But maybe if we break down this word a little bit, we'll remember better their meaning. So the I from at each word comes from the word immuno, which means that the technique we use, it's an antibody-based method to detect a specific protein or target. The H in immunohistochemistry comes from the word histo, which means that we will perform our immunostaining on tissues or 3D tissue models. Cyto, from in the middle of the ICC, comes from the word cyto. It means that we will perform our immunostaining on cells in monolayer or suspension of cells. Now, the C at the end of immunohistochemistry and immunocytochemistry comes from the word chemistry, which means that we will detect this signal using an enzymatic reaction. The F, the end of the word immunofluorescence, comes from fluorescence, which means that we will detect the signal using fluorescence. So now, hopefully, you will remember better what these words actually mean. So let's see how immunostaining works. I will just give a brief overview. So as I mentioned, immunostaining is an antibody-based method of detecting the protein of our interest. And the antibodies are macromolecules which are naturally produced by the immune system of an animal to defend itself against foreign matter, which we call antigen, because they don't belong to them. So let's assume that we have a protein which comes from human. We purify this protein, and we then, which we also call it antigen, and then we injected it in blue rabbit. So the rabbit's immune system will respond by producing antibodies. And the antibodies come in different flavors called isotypes. Mammals produce 5 different antibody isotypes: IgG, IgA, IgM, IgE, and IgD. And several of them have even subclasses. The most commonly used antibody in research is the IgG antibody. So let's assume that our rabbit immune system responded by producing IgG2A antibody. In this case, the IgG is the class and the 2A is the subclass. Now, the IgG antibodies consist of 2 identical subunits, each of them with having a heavy chain and a light chain, and these subunits are linked together by these sulfide bridges in a Y shape. Now, the part of the antibody that is antigen specific is called the FAB fragment, which stands for fragment antigen binding. and it will recognize the epitope, the part of the antigen to which it will antibody binds to. The FC fragment is species specific. It tells the rabbit that you produce this antibody. And instead, the FC fragment stands for fragment crystallizable region. So now how can we detect the antibodies? We can detect them either by direct labeling, where we will add a label to the FC part of the antibody, and then we can visualize it. Another method is the indirect labeling. How does this work? Again, we take our antigen, which we injected it into the blue rabbit, and the rabbit responded by producing the antibodies. Now, the FC part of the rabbit antibody is isolated, and then it's injected into a, let's say, green mouse. And the green mouse will respond, immune system will respond again by producing antibodies with an FAB fragment, which will recognize the FC part of the rabbit antibody, and with its own FC part, telling the mouse that it produced it. And now we label the FC parts of the mouse antibody, and then we can visualize our antigen. The part which is the antibody which is not labeled is called primary antibody. The antibody that is labeled is called secondary antibody. Now, how many types of antibodies we usually work with? We have monoclonal and polyclonal antibodies. Let's start with them. The polyclonal antibodies are produced by injecting the host animal, which can be rabbit, sheep, goat, chicken, with an antigen, which will lead to a immune response in these animals, and then the antibodies are extracted from the animal blood. In this mixture of antibodies, we have a mixture of antibodies which will all recognize different epitopes, but on the same antigen, which gives some advantages because we will have a higher overall antibody affinity against the antigen due to the recognition of multiple epitopes. We'll also have a higher sensitivity of detecting low quantities of the antigen, but also a higher chance for cross-reactivity and a relative high batch-to-batch variability. The monoclonal antibodies are generated in vitro in hybridoma B cells for mice. They are also available in rabbits. They will recognize only a single epitope on the antigen, which will give high specificity of this antibody, and thus low cross-reactivity, a low batch-to-batch variability, but takes time to produce and are more expensive, especially the one monoclonal from rabbits. We have also recombinant antibody, which are produced in vitro when we are cloning the heavy underlying chain in expression host, and then we are purifying the antibodies. They will only recognize a single epitope on the antigen, and they show very, very little batch-to-batch variability. Another types of antibody, in contrast to the IgGs, the IgG as antibodies can also give rise to antibody fragments, or can be used as antibody fragments if we are when we are incubating the antibody with different enzyme. We can get either monovalent FEB parts with only one antigen binding site, or bivalent FEB part which has two antigen binding sites. And when we are reusing them, well, we are using them if we want to block cross-reactivity. For example, if we are using primary antibody raised in the same host species, or we can block binding of secondary antibodies to Fc receptors, which could be present or are present in certain cell types or tissues which are infiltrated with these cell types. Or we can label them, for example, the FEB fragment with fluorophores. The good thing about these antibodies is that they are penetrating the tissue more effectively due to their smaller size. But the disadvantage is that we can only conjugate a few fluorophore dyes or enzymes to these fragments. Another type of antibodies are known as camelids or nanobodies, which in comparison with the classical antibodies, they lack the light chain, and they were originally identified in camels, but later on they were found also in llamas, alpacas, and in some shark families. The variable part of the antibody is quite small and have very special finger-like protrusions. And this little part of the variable part can be isolated, and it's they are called nanobodies, and they can also be cloned. Again, why use nanobodies? Again, because they're penetrating more effectively the tissue or the sample due to their smaller size and these extra long finger-like extensions. They are more stable at room temperature and can be easily cloned. And easily tagged with fluorophores. Of course, then we have to be very careful when and choose the correct primary and secondary antibody pairs. That is very important, and especially if when we will perform and want to detect several antigens on the same sample. So the secondary antibody must be raised against the host species of the primary antibody, and it must be specific to the isotype class of the primary antibody. If we are working with FEB fragments, we can only select the anti-FEB fragment secondary antibody or the full length, but not binding to the Fc part of the secondary antibody, because that's not there. So here I give an example. Let's say that we want to identify antigen 1, 2, and 3 on a human tissue, and the antibody binding to antigen 1 was raised in rabbit. That means that the secondary would be raised in another species, but against the rabbit primary antibody. So, for example, donkey anti-rabbit. If the antigen 2, the antibody was raised in mouse, the secondary should be, again, as an example, donkey anti-mouse. For the antigen 3, which the primary was raised in goat, with the goat anti-antigen 3, the secondary should be a donkey anti-goat. So please pay attention when you are selecting your pair primary secondary antibodies. Now, we said that how we can visualize our target. There are two main techniques, chromogenic and fluorescence detection, which can be applied regardless if we are performing a direct or indirect labeling. And let's see what's the difference between them. So when we are performing chromogenic labeling, the antibody, either directly or indirectly, will be labeled with an enzyme. The most commonly used enzymes are horseradish peroxidase or alkaline phosphatase, but probably there are a few more. And after we incubated our antibody, the enzyme labeled antibody with our sample, we will add a colored substrate, which is called the chromogenic dye. For example, DAB, which is one of the mostly used. And at the site where the substrate will encounter the enzyme, a precipitate will form. And below you can see a very beautiful image of a chromogenic staining. Fluorescent labeling. In this case, our antibody would be labeled with a fluorophore, which absorb and emits light, and we can visualize it under the when shining light on our sample. But when should we choose what? When should we choose a chromogenic labeling or a fluorescent labeling? When we want to visualize chromogenic labeling, for that we required color cameras, and these are less sensitive than black and white cameras. Also, if we would like to use or to detect the different antigen, we have to be very careful that they are localized in different parts within the cells, so their localization should not overlap. We also have to keep in mind that contrast, sharpness, and resolution are lower than in fluorescence. But the good thing about it is actually that we don't have to bother about the autofluorescence because we won't see it. But sometimes the tissue can show some color, especially if the fixation was performed by very old fixatives or glutaraldehyde. Another positive aspect of chromogenic labeling is that the labels are preserved much longer. If we perform fluorescent labeling, well, we can also image it with the color cameras, but mostly we are using black and white cameras, scientific black and white cameras, because they are more sensitive than colored cameras. It's possible to label more molecules at once, even with overlapping antigen localization. Also, we will get a better contrast, better resolution in our images. But unfortunately, when performing fluorescent labeling, we have to deal with autofluorescence. And if we didn't do our homework to check before how we match our fluorophores with the specification of our microscope, we will have a risk of bleed-through between fluorescent dyes. Another negative aspect is that these fluorescent dyes can fade during imaging and storage, so we cannot preserve the sample as long as when performing chromogenic labeling. So now that we have an idea of how immunostaining works, let's see where should we look for our antibodies and what antibody validation is all about. So I will again make an attempt to start a voting pool if it's possible. Obviously, I have problems with it. Let's see. So can we have a little voting here? And I have two questions. Before you purchase your antibody, do you check if the antibody was validated? And the second question is, do you validate the antibodies you are using? Okay. Let's hope that I can explain and make you understand why antibody validation is important and what.
I think you need to share maybe you need to share the results for people to see. I'm not sure.
Can you see the voting at all?
No, we cannot.
We can see the voting, but we never saw the results yet.
Need to share results. Yes, exactly.
Can you
You
see
can
now?
comment on the results, maybe comment a little bit on the results so that people.
So as I can see, not not too many of you actually check if any validation data is if you check if the antibody was validated, which very much could have been, but you didn't pay attention to actually
28% did check. So
yeah,
I think
they check,
most people check. Yeah, that's pretty good.
which is good. And quite a bit, many of you also validate your antibodies.
It'd be interesting to know what you guys do. Maybe we'll have to discuss that together.
Okay, can I continue?
Yes.
So what is a good antibody? A good antibody should fulfill the following criteria. Specificity, which is one of the most crucial issues, and it means that it should only detect the target antigen. Sensitivity, that it will detect low amount of the antigen, and reproducibility, that the same level of specificity and sensitivity will be performed with the same level of specificity and sensitivity across batches and throughout your whole experiment. But where will you find such an antibody which will fulfill all these requirements? First of all, it's quite important. It's important to remember that it takes time. That you should take your time when choosing an antibody, because sometimes finding the right antibody is like finding a needle in the haystack. There are many places to look for, but you also have to be very careful when choosing the antibody, because 50% of the commercially available antibodies are either not working or might not fit for the purpose of your experiment. But you have to start somewhere. Of course, most of us start by looking to antibody citation or scientific publication, and which in general gives a very good overview of which antibodies are available or have been used by other researchers. But unfortunately, just that an antibody, the fact that an antibody has been cited in many journals, does not necessarily mean that is the best one for your application. And that it actually was carefully used in the experiment. For example, that enough controls were performed, and the images that they provide actually shows correctly or include all the controls and data. So, and also to look through all the journals and publication to find this information manually is time consuming, and sometimes you might miss some information, or you just don't pay attention. Another place I suggest that you should look for, and sometimes even start with, is the Human Protein Atlas, where you should go to learn more about your protein of interest. This database has a tremendous amount of information regarding proteins due to their multitude of samples they characterize with antibody-based assays. So it will give you a very good idea and information how what is your protein, how it should look like in different assays, how they perform these assays, so you can continue searching for your antibody. And there are many antibody searching engines that you could take help from. which are comprehensive databases with millions of antibodies, where you can find or you can search for your antibody of interest. And here, which will spare you lots of times. It also will bring together many vendors, all selling or having an antibody of your interest. So basically, you will spare a lot of time. And also, here I only show a few of these search engines. For example, antibody registry, BioCompare, Antibodypedia, and probably many of you already are using these search engines. And of course, they are not all the same. They differ quite a lot, not only by the number of antibodies they have in their database, but also they differ by what kind of information they provide in these databases and the filtering options linked to each antibody. Some of them, they have direct links to publication and application. Some of them even display the material methods where this particular certain the antibody was used. Others also include user reviews. Some of them identify if the same antibody is, if you are actually looking at the same antibody with but sold from different vendors under different catalog numbers. And probably one of the most important information, they tell you if the antibody was validated, or and the validation was done by the supplier, or they can also provide independent antibody validation data. So they are very useful search engines when looking for antibodies. Another search engine which I like very much is the research resource identification portal, which you can find at it's hosted by the SciSbranch data homepage, and was created to improve the reproducibility in science. So what is this database all about? So basically, it's again a pool, a central location from many community databases. And here you can look for not only antibody for that matter, we can look for cells and organisms. And all of these have a unique RRID number, which stands for Unique Research Resource Identifier. So you can basically search using this number to find your antibody. And many many publications or journals request in the material and methods that you write down the RRID number of the antibody you use. So when you are doing a search in the RRID database, you will gain tremendous amount of information. But first of all, it will take you the correct source of the antibody. It can give you so you can track the history of the antibody, who is selling it, who produced it. You'll find information regarding problems with these antibodies. In some cases, you will even be able to contact the researchers who use these antibodies, and which will probably help you in troubleshooting your own experimental data or experiment. So I urge you to take a little time and go through some of these search engines and try to find which actually works for you and get the best out of them. So if we think, let's find that we find our antibody. We think that this antibody will work for my experiment. You go to the vendor site. The first thing you should look at is the data sheet. First of all, do they provide the data sheet? Because the availability of a data sheet is a must. A nearly empty data sheet, it's a strong indication that that antibody was not really validated. They don't know too much about it. But what should be available in a data sheet? First of all, the application field. What can I use this antibody? In which application? Western blot, immunostaining, ELISA, immunoprecipitation. If you don't know with the application field, or you get very poor information regarding it, you are not happy with the images or with the Western blot that they provide, or you don't get any validation for your particular application field, then you don't buy it, or you have to validate it yourself. And of course, the manufacturer cannot validate all the antibodies for your specific application and for your specific sample. So that's why we have to be well aware of it and decide, if I buy it, then I have to do it. Don't take chances. Another information should be the clone number, or a commercial product number, antibody ID, or, as I mentioned, there are numbers, so you can always refer back to it, and you can search for it. What type of antibody is it? A polyclonal, a monoclonal, a recombinant, or what type of isotype, class, and subclass. If you don't know this, it will be very hard to assess your results, and also to design, for example, what kind of secondary antibody you should use. Clear definition of the immunogen. Was this the antibody raised against peptide or protein fragments or full-length protein? Again, if you don't know this information, you will have a hard time to decide if the antibody is specific enough or not, or if you don't get, for example, in Western blot band, to understand, yeah, well, of course not, because it was raised against the full-length protein, and which has a 3D structure. The cross-reactivity. The more cross-reactivity data you have, you will get. You will also, it will tell you, you can evaluate if you can actually use it for your application, but will also show that the manufacturer or the vendor actually made an effort and validated the antibody. Antibody epitope affinity. Is it has a low or high affinity binding? Because if this is not specified and that antibody actually has a low affinity, well, you will get weak signals, high probability, high background, which will also give rise to low reproducibility in your experiments, but also will cost you quite much more, because what you will inevitably do, you will just increase more and more the amount of antibody you use. The concentration of the antibody. What additives and buffers? What is in that solution with the antibody comes with? First of all, the concentration is important, so you are able to if you don't have the concentration, that will prevent you from protocol optimization. And some of the additives, if they're not mentioned, you will not be able to know if you can use this particular antibody when you want to conjugate it with fluorophores, as example, azide, or interferes with quite a few processes. And storage. Considering the high cost of many antibodies, stability is very important. And you should always, if you don't know how to store the antibody, that will influence its stability. The antibody will degrade in time, and it will influence the reproducibility of your data. So recommendation about storage is a must. And validation data. How did they validate the antibody? What type of sample they use when they validated the antibody, and what methods did they use for validation? So we we will continue with how to validate antibody and what antibody validation is all about. But let's take 2 minutes break to drink some water, and then we can continue with antibody validation. Okay?
So back at 39.
Yeah.
Thank you.
Okay, so let's continue with antibody validation. What is that all about? So antibody validation is the experimental proof that an antibody is suitable for the intended purpose. And that is that the antibody will specifically detect only the antigen of interest, and it will work in the desired application, and has a consistent performance, meaning the results are reproducible. Now, a committee, an international working group for anti-validation, actually came up with some recommendation standards for antibody validation. I highly recommend the paper by Ulen, which came out in 2016, where they outline the 5 pillars for validation strategies that you should use when you want to validate your antibodies. And these are genetic approach, orthogonal approach, independent antibody approach, tag protein expression, immunoprecipitation, and mass spectrometry. And all of these, each of them have their strengths. And in this paper, they very nicely go through all these pillars and what is the criteria for then you are using each of these strategies. and which applications are suitable for when using them. So an antibody should be validated with at least one. This is absolute minimum of the conception pillar, which are listed above for a specific application and antibody. And most of the antibody manufacturers are actually, here I only highlight a few of them. They are very clearly stating how they validate their antibody. So it's quite important that you pay attention and you look for validation data in the data sheet. Now, when should you as a researcher validate an antibody? Well, if the vendor did not validate the antibody for your type of sample and your application, if the vendor did not provide extensive validation data and data specific for your application in the way you are expected that the data should be presented, meaning so I would say unsatisfactory validation data, which can also be in publication or at the vendor side. For example, not a Western blot or not visible controls in the Western blot, no controls presented then in the immunostaining and so on. Or when you are producing your own antibodies. And here I just have a list of a few of the guidelines out there which you can follow, so you understand and you know how to perform validation of an antibody. Now, among all these validation pillars, and there are a few that are widely used for example, like Western blot to show specificity, to characterize some of the important characteristics for an antibody. Western blot is highly used to show specificity because it's easy and it's also easy and cheap to perform. But you have to keep in mind that a Western blot will only be informing you that the antibody is able to bind to the denatured protein. So if the antibody was raised against folded protein, and here we can give as example the activated integrins, the Western blot will not cannot be used, so you will not get anything. So also then you perform a Western blot. Let's say that the vendor shows a Western blot, but is definitely not in the cell or tissue type that you are working with. Then you have to remember that you have to perform a Western blot using extras of your own sample. You always have to include positive and negative controls, knockdowns, knockouts, overexpression or expression in the cells, or when you work with tissue known to express or not the pure protein of interest. And keep in mind that proper design control is holding the key to conform specificity. Without controls, it's your method, it's not really, it's invalid. So when you run a Western blot, you are expected to get one band at the correct molecular weight, which will confirm that the antibody binds to the protein of the expected molecular weight. But without controls, you will never be sure. Sometimes you can also get several bands, and this is due to protein modification, proteolysis, or presence of splice variants, but can also show unspecificity of your antibody, like for example in this blot, where you can see that in two different cell lines you can have some extra one that should not be there. Sometimes due to the low relative concentration of the target protein, basically you might not identify any band of interest. Now, If we are using genetic methods also to show specificity, which is a very powerful tool to validate candidate antibody, especially for immunostaining. You will use knockouts, knockdowns, or overexpression. It's quite easy to achieve in cells, where we can increase or decrease or remove the amount of the protein your influence and compare the results. But unfortunately, then working with tissue, we are very limited with the number of knockdowns and knockouts available in animal models. To produce transgenic animal takes time, and some genes cannot be knocked out because they will lead to low cell viability. And the knockdowns are generally transient and rarely 100% effective. You can use indifferent antibody approaches when you can use 2 or 3 independent antibody, which will target different epitopes on your target protein. And it should be a significant correlation between the expression of the expression level then using all these antibodies. You can use tag protein expression. You can tag your protein with either a peptide or fluorescent protein, and you should get the same pattern when visualizing your signal. But you also have to, don't forget to include controls with only the tag, so to avoid the potential artifacts which are induced by the tag itself. Affinity of an antibody, it's will tell you how strongly the antibody binds to the antigen, and is defined by this Ka or KD, Ka association constant, or KD dissociation constant. And this is actually not always provided in the data sheet, but basically you can have high affinity antibody, which will quickly and strongly bind to the antigen. They show great sensitivity, and they will maintain the binding to the antigen even in the more stringent conditions. If you have low affinity antibody, will quickly bind to the antigen and have low sensitivity. Now, if we go back to what I mean by stringent conditions, basically that means that we make it difficult for the antibody to bind. With how you do that, if you want to check or to see that your antibody has a high affinity, you would, although it sounds counterintuitive, you will use it under stringent conditions, meaning that you will lower the concentration of antibody. You incubate at 4 degrees, which usually or you incubate it over longer time, usually overnight. This will also increase your the signal and will lower your background. Unfortunately, if you get a low, when getting low specific stainings in your sample, but you get some stains outside the sample, my advice is to decrease the primary antibody concentration, ergo to increase its stringency. This will, as I said, will improve the sensitivity during the imaging. And I also suggest first that you make dilution series, and also to increase the low specificity, you should check that your mounting medium is not autofluorescent. Reproducibility between experiments, it's very important, not only between the experiments, but also between batches. You have to, if you check the reproducibility within your current batch that you are using, my suggestion is to run the same immunostaining protocol in 3 different days, and then you compare your results. To compare the reproducibility between batches, first of all, buy a new batch well before the first one runs out, so you can compare with the one that you are actually using already. You have a much higher risk and lower reproducibility between batches if you are working with polyclonal antibodies, so which are thus a much poor choice of antibody if you are performing long-term experiments, or you perform experiments that have low tolerance for variability. But if this is the only antibody you can use, then you buy a new batch, ask for the same lot. Reproducibility is lower when you are using monoclonal antibodies. The disadvantage is that if you have low abundance protein, you might run out the risk that you cannot detect it. Or if you are looking at proteins with epitope variability, for example, proteins with showing translational modification or conformational change. The recombinant antibodies are the best, show the less variability between batches, but they are quite expensive. Always contact the company and sometimes they complain if the new batch does not perform as well as the previous one, or you need to increase the concentration a lot between the batch you already have and the one that you ordered. So the reproducibility will also be highly influenced by how the antibody was stored, because antibodies also degrade in time, especially if they are not stored according to the manufacturer recommendation. Of course, you should not forget to validate your secondary antibodies, because they must satisfy the same standard as primary antibodies: specificity, affinity, sensitivity, and reproducibility. And here I can show an example of less reproducibility of a secondary antibody between 2 batches. And as I mentioned, that secondary antibody must be raised against the host species of the primary antibody. Always have a control sample, meaning no primary or secondary. So you can assess the cross-reactivity with endogenous IgGs or serum IgGs. Always, then you order a secondary, demand the full spectra of the fluoroform bound to the secondary antibody, not only the maximum of its excitation and emission. And now, by now, you should know why. Be aware that the same antibody may be sold under the different names by different vendors. And it will be really tremendous waste of time if you will have to test. You will think that you bought a new antibody, but actually you are using the same. And there are signs that you can might help you to figure out if it's the same antibody. For example, the same formulation of the buffer. If you see the same Western blot or immunoprecipitation or same images in Western blot IP or immunostaining, if you have the same vial size, quantity, and dilution, you have the same recommended protocol, the same reference list, this should already make you think. Then you go and you take your old data sheet and you compare very carefully. They are the same, so actually I'm ordering the same antibody, and the price actually doesn't make really a difference. That will not indicate that it's a different antibody. So always work with company that will work for you, that will help you to troubleshoot the antibody if it doesn't perform as expected. So let's have another 2 minutes break so I can, and I will start with my last part of my talk. Okay?
Oh, 57?
Yeah.
Okay, thanks.
So let's continue with the last part of my lecture. And we will continue with the controls. Which are the essential controls needed in performing an immunostaining? The controls are a must because they are the requirement for robust data interpretation. And the control sample must be processed in exactly the same way as your main sample. And you should always keep in mind that you have to have controls for each new antibody you are using. So the controls that are required when performing immunostaining are the no primary control, basically no primary, only secondary antibodies. And this control is needed to determine if the secondary antibody is binding non-specifically to your cellular components. And you should not get a signal when visualizing your sample. Sometimes you might get some bright dots looking at debris, hopefully both in your sample, but also outside your sample. And I would say that those are secondary antibody aggregates, which that occur, aggregation can occur due to improper storage conditions. And this is when I recommend that you take the vial of the antibody, you spin it down. First of all, you vortex first, you spin it down, and you just take the supernet and then using it. The autofluorescent control, and we talked today already about the autofluorescence. And this control means no primary and no secondary antibodies, and is needed to determine if the observed fluorescence is coming from a background fluorescence. Of course, you should not get a signal when visualizing the samples. And if you get a sample, that means that your sample is autofluorescent and you need to treat your sample. And you are I already mentioned the ways you can do that. The expression controls, either positive controls when you overexpress your target, or you use a sample, especially in the case of tissues, not to express the protein of interest, and you should get a staining, and the staining should be as expected. If you do not get a staining, either is something wrong with the staining protocol or with the antibody, might have not been validated. So you have to do some extra validation steps. For a negative control, knockdowns or knockouts of a sample, not expressing, so they don't express the protein of interest. No signal should be detected. Of course, this is much difficult if you have tissues. What you can do is to perform a knockdown or knockout in cells, and then make a pellet and work with that pellet, process it as it should be a tissue. If you do get a staining, that means the antibody is not specific. As isotype controls, when should you perform that? If the tissue is involved in immunity, so likely to have Fc receptors on the surface, or when the tissues are highly infiltrated with immune cells, there are these type of tissues too, or if the antibody is raised in the same species as your tissue. And of course, no signal should be detected. Now that we know which control, so when we prepare our sample, we know how many, and we split our cells, how many wells with the cells we should have, how we use with tissues, how many tissue sections we have to cut. Now we start to proceed with the immunostaining steps. I will not, and I will highlight only a few of them in this lecture. We already talked about fixation. I will just say the most important message. And when fixing, to find that the fixative will work with your antibody, this means that you should follow the recommendation on the antibody supplier. Or use a fresh fixative solution when fixing your sample. Optimize the fixation time depending on your sample thickness, meaning cells or tissues. Do not overfix or underfix, and wash your sample gently and thoroughly after fixation. Now, quite often, we are looking for our target protein is inside the cell, so we have to allow the antibody to enter the cell, so it can reach its target. So the permeabilization is used just to make holes in the membrane to allow the antibody to enter. And there are two types of reagents, detergents or organic solvents. In the detergent family, we have mild detergents like saponin, gititonin, 320. They would only permeabilize membranes containing cholesterol. And quite often the detergent must be included in all solutions during the entire labeling procedure. We have Triton X100 and NP40 belonging to a much harsher detergent. They permeabilize all membranes. Now, in the organic solvents, they act themselves as permeabilization agents, especially methanol and acetone. By dissolving the lipids, this is how they make a hole in the membranes. You should always perform the permeabilization on ice, and you have to be very careful that due to their being harsh solvents, they can damage fine structures, like example microtubules or even other organelles. So always use a permeabilization engine that is suited for your scientific question, because if you use the wrong one, you might actually remove your target. So just asking your friend, what do you use? Your friend working in a totally different samples and condition might give you the wrong answer, and you would remove your target. Always you have to adjust the concentration and time depending on the thickness of the tissue, even for permeabilization. And of course, if you are using organic solvents, you have to make sure that the antibodies are compatible with it. When we should perform antigen retrieval, mainly then when we are using formalin, PFA and formalin fixation, paraffin embedded samples. And the role of the antigen retriever is to break the crusting that were generated by fixation and to unmask the epitopes. And it's always performed before the staining. We have two methods, a heat-induced method, where we work with very high temperature, either using pressure cooker, microwaves, and water bath, and different citrate, different buffers. The most commonly used are electricity and citrate buffers. This method is prone to optimization. You have to, because it's sensitive to pH, time, temperature, and buffer conditions, and each of them will depend on the tissue, the fixative, and the antigen. And the negative part when using antigen retriever is that the sample may detach from the cover slip, so you have to incubate them horizontally in the buffer. Another method is the enzyme-induced. It's less used, but what it does is that will cleave the peptides that are masking the epitopes. Here also optimization is needed, concentration of the enzyme and length of the treatment. It's much prone to destroy the tissue and to remove the antigen. Can you avoid antigen retrieval? You can give it a try, definitely, before starting this method. For example, you can dilute the antibody at different pHs or cation concentration, different dilution and incubation times. You can also choose a polyclonal antibody instead of monoclonal. antibody, or you can use nanobody. But of course, if nothing of this works, you have to do antigen retrieval. And here I just have an example of how by changing the buffer and the buffer concentrations give you a very nice staining for two different antibodies, one of them working low pH and both in low pH and high pH, depends on the buffer, and the cadherin antibody only working at high pH. So you have to make some optimization when performing antigen retrieval. The blocking, that what is that used for? It says to complete the specific binding of the antibodies, and it's of course done before adding the primary antibody. We have several blocking reagents. Usually is the normal serum, which is you have to use the serum from the host of the labeled antibody. If you have several antibody, you have to make a mixture of blocking sera for all the against all the species of your secondary antibodies. You can use protein buffers. Most commonly used are BSA, non-fat milk, and gelatin. Also here you have to pay attention that, for example, the BSA is free of IgGs, so you avoid cross reaction with the secondary antibodies, which will increase your background. And if you use non-dry fat milk, it's also you have to read the recommendation because it's not really used when you want to detect phosphorylated proteins. Should not be used in assays which include biotin binding assays. You can also use FAB fragments, which are used to block endogenous immunoglobulins in tissue section or on the cell surface. Then, for example, your antibody is raised in the same species as the sample. And here I have an example. You have a mouse tissue where you want to detect antigen G, but it also has endogenous IgGs. And now, and you want to use a mouse antigen X antibody. This is when you should use an FAB fragment to block the binding sites of the endogenous IgG, so you can add your mouse antigen X antibody and the corresponding secondary. Don't forget that even from chromogenic labeling, you have to perform blocking, and especially if the tissue shows endogenous enzymatic activities, and you are using HPR or post-peroxide oxidase or alkaline phosphatase detection, and in some cases in biotin-based detection system. Always try different blocking agents because it will greatly help you to reduce the background and improve your signal to background ratio. Always use the same blocking buffer for diluting the antibody that was used for the blocking steps. So yeah, you have the best, you have the optimal assay conditions. The washing steps should not be ignored. Always perform them after each step in the protocol. You have to wash a lot, but gentle, and use the common buffers are PBS or PBST, TBS or TBST, but and the most important thing is that you are quite often the manufacturer are specifically tell you which buffer you should use because this works best with their antibody. So washing steps should not be ignored. And in my last slide, I will just tell you a few things about antibody penetration in large samples. The large samples, we can consider organs, large organoids, thick tissue slices, they require extra attention before, during, and after the immunostaining procedure. You should always assess if you have an even distribution of your staining through the whole sample. So basically, you should be critical with acquired images. If you look at image is acquired and you are imaging a big sample, ask yourself, can it be that there is no staging in the middle of my stack? Can it be this be biology or something else? Why is my nuclear staining weaker in the middle of my sample? Why do I always get staining, my staining so bright at the outskirts of my sample, but not in the middle of my sample? That could be antibody penetration issues. Basically, the antibody didn't reach deep inside your sample. And that depends, of course, on the tissue types. Some of them are more dense and they had the ACMs, which make hard for the antibody to diffuse through. Or the antibody will bind to the first layer of the uncounted epitope, so it's not too much left to diffuse into the sample. But there are solutions to that, and there are many protocols available which will help you to optimize this antibody penetration in the samples. So you can, you will have an even distribution of your antibody or your whole sample. Here I only mention a few of them. For example, the switch on and off protocol, where you can just switch the buffers between. So basically, you allow the antibody first. You incubate your big sample with the antibody, but the antibody will not bind to anything, but will just freely diffuse through the whole sample. And after a while, you change the salt concentration, and then you allow the antibody to bind. And now being diffused through the whole sample, it will bind everywhere. Other application are, you can use an electromagnetic field, for example, the E-Tango protocol, or you can apply pressure, and I'm pretty sure there are many more out there. Thank you for your attention.
Hi, Abby. Thank you. Okay, so
Almost in time, sorry.
that was a lot of information. I would like to know if you have any thoughts or comments about all this compared to how you prepare your antibody. Ignore.
Hi. So yes, of course, it was a bit overwhelming to. It's kind of like list of things to do when you go back to lab after this course. But I actually wanted to ask two questions regarding the first, the human protein atlas antibodies. The thing is, I know that like during this human protein atlas, like them trying to like having this atlas, they ended up developing some of the antibodies themselves, and then now they founded this atlas antibodies and they are like selling them as well. But I was curious about your opinion about like the quality of those antibodies, if you have any input in that. That's my first question.
Personally, not in the sense that I never bought anything, but if I talk with my colleagues or scientists or researchers, they have a very good experience with those antibodies, because actually if they are for the purpose they are validated for, they perform very well. So the data and the validation data for that particular application, for that particular cell line, works very well. And this is why I said that means that if you think, yeah, but it worked there, so if you buy it, now you have to try it for your own sample, because of course it cannot be used for everything. But what I heard, and our users and other of my colleagues, so to say, who buy from there, or they use their antibodies, they were quite happy.
Do you have any experience now, or is anyone else about these antibodies?
I think I have one in my list that I used from them, and I'm like particularly fine with it. But in general, I didn't use much. So if
Anyone else has used the protein atlas antibodies? Doesn't sound like it.
Yeah. I mean, now they go with the brand Atlas antibodies, if anyone is not sure, maybe.
Which one is that? Yeah.
Yeah, they quite improved their homepage lately. It's
Yeah.
a
Maria?
brand new one.
Oh, Sadia says she used and for her it didn't work.
Right. So, Maria, did you have also some experience?
No, I have another question.
Okay, sorry. Okay, so maybe it depends on the antibody as well, and clearly it doesn't mean because they have tested an antibody. They do test it on quite a number of cell lines, but
And
it might be different
applications.
in tissues as well. So, Maria, yes? Oh, Ilknur, you had another question, right?
Yes, thank you very much. Can I just quickly ask that as well?
Yes.
So the second question is, what do you think about overnight? How long is overnight incubation? Because like primary antibody incubation, we do this overnight, and one of our protocols has overnight fixation in 4°. And when it comes to fixation, we would like to be a little bit more specific because it might change a lot of things and might mask the tissue, and we don't want that. But in that case, how long overnight is for you?
No, for
Overnight
antibody or
fixation
for
in 4
fixation?
degrees. Yeah. Overnight fixation. Is it in the tissue or is not cells, right?
Yeah.
So this is the protocol that is recommended by the manufacturer of your antibody?
It's the like tissue. It's the protocol that was like done by. It's kind of like the protocol for the generation of the organoids, and this is what they were doing from the tissue. It's a
Yeah, this is published somewhere.
Yeah, it is published, and we are trying to like recapitulate what they have done. But when you say overnight, it's a bit vague, and we were thinking maybe 8 to 10 hours is good.
Yeah, in general, I mean, depends quite a lot of the size of your of the sample. That's what it's all about. You have to basically optimize depending on the size. Not so much to increase the concentration or so, but the size itself will make a difference. So it's not too. How big are your samples?
Let's say like 1 centimeter tissue pieces.
Yeah,
Sometimes.
so I think if you're worried that you're overfixing it, for example, which is the main issue, I think you could try, for example, 12 hours and 8 hours, something like this, and see if you see a difference in the and 4 hours or something like this. You can try. Plus, you can also cut the tissue a bit smaller and then see, and you compare clearly with what is published in the literature to see what to expect. So if after 12 hours of fixation, you do not see the localization, the protein localization that is expected, this is when you'd try something else, basically.
Thank you very much. Thanks for your input.
Maria?
Yeah, I have a question. How could you do the antibody validation on biopsies? If my sample are biopsies from surgeons, how can I do the antibody validation there? Like which method is the most optimal, and at the same time I don't waste the sample.
You don't know what? Where is the sample? Or you don't know?
That I don't waste.
You don't want to waste the sample. Yeah,
I
clearly.
get
Yes.
a
Yes.
limited amount of slides. Yeah.
But then you bought the antibody. That should have been the first step to see was it, for example, validated in human tissues, or for example, you already get a little clue if it might work. It's not easy if you are very limited in the amount of, but then at least then you try to perform when you perform the immunostaining, you perform the controls. So you try to have some the controls, so you know at least to some extent how it works. It's very difficult, and you are limited in the amount. How many slides do you have?
Yeah, this
How
is
many
hypothetical.
antibody do you have?
This is hypothetical because I don't do immunostainings, but I do in situ sequencing, so I don't do immunostainings. But in the case that I would like to do a immunostaining after the in situ sequencing, yeah.
And then you will definitely, what you will validate this by running all the controls that are necessary.
Yeah, but which are those?
No
controls?
primary, no secondary, only secondary, depending on the type of the tissue you have. Human tissue might need to perform the isotype control, and then you see the results comparing with the results that then you are imaging your main sample.
Okay, so no primary and no secondary.
So no primary, no secondary. That will be to reveal the level of the autofluorescence you have in your tissue, and probably that would be the first that I would do. So to know actually after I perform my immunostaining, I do need to take care of the autofluorescence. And usually, depending on the type tissue, but human tissue are quite autofluorescent, brain, liver, like a Christmas tree. Then you have no primary, only secondary. It will tell you, is my secondary antibody binding specifically? But of course, if you use a directly bound primary antibody, then this control, you don't need to have it. What type of tissue? For example, some of the lung tissues are highly immunogenic, so they contain cells. So they might be infiltrated at a certain cell time that will present IgGs at the surface. So you then need to do some isotype controls, for example. So these are, something that you always have you have to perform, that you are starting to work with a new tissue. It's a must. Otherwise, you will never know when you look at the results, what is actually I'm seeing there.
I think autofluorescence and to see if the secondary binds as specifically or the primary binds as specifically. So isotype control and no primary. These are sort of a must for every tissue or every sample and every antibody. This is really important so that you don't have something, you're not imaging something that binds to the wrong place or that autofluoresces. This is really the minimum that you can do if your sample is very precious.
Okay, thank you.
Are there any more questions or comments about, yes, Yubei?
Yes, and Gabby, you mentioned that one of the reasons we permeabilize our samples is to get the antibodies into the cells. But what if we're interested in the extracellular matrix proteins or cell membranes? In this case, should we or can we skip?
If it's the outer is the extracellular, so the outer part, it can be plasma membrane. It can be bound to inside or the outside, and you don't have to do. You shouldn't permeabilize. But sometimes you want to look to several things, both outside and inside. Then I suggest that first you incubate with your antibody, which will bind to the outer proteins on the plasma membrane, and you do everything. Then you wash thoroughly, so you don't leave any trace. Only the stringency I was talking about, that you really make the antibody bind, and only after that you will perform a permeabilization. For example, if you look for your target is inside the cell. You understand? Then now, once the antibody is bound, it sticks there. Is everything goes well, so that you don't have to care about. And then you do the wash, and then you add the permeabilization, and you incubate now with a secondary antibody, which will recognize something inside the cell, if that's what you want, or I don't know. So, but always the first is the one you don't have to. That's why the solvents are very critical to use them or not, because they, permeabilize everything. Maybe the nuclear membrane is a little bit tougher, but otherwise it makes like big holes everywhere. Now you can lose your antigen.
I think it also depends on how thick your antibody is, because even if so first, if you have a membrane protein is your target, you have to check if the antibody was raised against the extracellular side of the protein or the intracellular side, because that can be the case. And then also, if your sample is quite thick, the antibody will have to go through number of cells. So it may not diffuse if you do not permeabilize. So it also depends on the type of sample you have. When you have like a cell layer, like a monolayer, you can use and you know that your antibody is against the protein that is on the outside of the cell. You can use no permeabilization, or if you have floating cells like blood cells. But if you have a tissue and is thick, then you might have to use permeabilization anyway, even if the protein is recognized as outside.
And then would you call 7 or 8 micrometers thick?
7 or 8 micrometers, you are cutting every single cell. So I think that is thin enough that you can try without.
Okay, thank you.
Yeah, that you can try without. But...
Yeah.
Thank you.
Anyone else? So we had a little something. Sadia, yes, you have a site antibody. What was that? Can you comment, Sadia, on if you can turn on your mic instead?
Yes, hi.
Yes.
So the thing is, this site AB website, it's very good. You can type in your antibodies, and then it can give you features like which dilutions people have used. So it's like a library they're building for different protocols for Western blot, for immunostaining, for tissue staining. So you can get the antibody validation or like concentration, and also get the references. So it's very good.
Very good. So this is one of these libraries that Gabby was talking about. It's very good.
Yes.
It's
Yes.
definitely worth to go take a little time and go through of them. Not might everything work for you, for yours, but you can find the one that actually works for you. And you gain a lot of time. And you will learn quite a lot regarding your antibody application and so on and so forth. Which publication you can look at, which not, because the data is not reliable. It's really very nice.
Good. If there are no more questions, we're going to go back to the Zoom, the workshop Zoom, and then we'll have a little discussion. You can fill in the 5.2